Ајде.
Добра вечер или добро утро за Америка. Океј, добра вечер или добро утро за Америка. Добре дојдовте на 23-тиот вебинар на Македонското научно здружение за аутизам поред. Денеска ќе имаме исклучително интересна гостинка од САД. Тоа е професорката доктор Џуди Ватер. Пред да ја дадам збор, ќе го прочитам нејзиното кратко CV или кратка биографија. Се надевам дека ќе ви се допадне таа исклучителна стручњак имунолог во нејзината професија. Инаку моја пријателка скоро 20 на години преку интернет. Таа е вработена на одделот за интерна медицина на Универзитетот во Калифорнија Дејвис уште од 1999 година. Во 2000 се приклучи на факултетот на новоформираниот Универзитет of Калифорнија Дејвис Mind институтот и каде што го започна истражувањата од имунобиологијата на аутизмот. Доктор Ватер спроведува програми за истражување кои се однесуваат на автоимуни и клинички имунолошки нарушувања, вклучувајќи ги биолошките аспекти на нарушувањата од спектарот на аутизмот и ова вклучува разбирање на улогата на мајчинскиот имунолошки систем за време на бременоста во здрава и променета невроразвојна средина. Примената на имунопатолошката позадина на доктор Ватер беше клучна во дисекцијата на имуните аномалии забележани кај некои индивидуи со аутизам и во диференцијацијата на различните фенотипови на однесување на аутизмот на биолошко ниво. Секако, таа е вонреден директор за биолошки науки на Mind институтот и директор на Дејвис Mind институтот, Центар за истражување на интелектуални и развојни пропречености. Таа исто така служеше како директор на Центарот за детско еколошко здравје, финансиран од Универзитетот во Калифорнија Дејвис, истражувајќи ги потенцијалните фактори на ризик на животната средина кои предонесуваат за инциденцата и сериозноста на детскиот аутизам. Значи, она е доктор по имунологија на Универзитетот Калифорнија Дејвис. Денеска, како што кажав, ќе зборува и како што видовте деновиве на нашите памфлети за гестацијското влијание врз причините за аутизам. Денеска само Македонското научно здружение за аутизам учествува во ова, така што нема да имате превод симултан, особено некој од вас на минатите вебинари бара оригинал. Сега ќе ги имате во оригинал слайдовите на англиски, ќе го имате нејзиниот глас во оригинал и после тоа ќе отвориме широка дискусија. Може би ја од време на време нешто во врска со МНЗА и нашите активности да споделувам во четот, а вие исклучете ги вашите камери и микрофони, така што на крај, ако треба, ќе се вклучите во живо или можете во questions and answers да ги поставувате вашите прашања. Сега ќе преминам на англиски. Dear Professor Џуди, Dear Professor Џуди, I introduced you on Macedonian, your short bio. I told to Macedonian professionals and parents that we had around 20 years friendship and we exchange knowledge. And I'm very thankful on the very beginning of the 2000s when you sent me some articles of you which helped me in my PhD. That was in 1999 until 2002, until 2002, and after that we have exchanged some experiences, and thank you for your support of me and also of Macedonian Scientific Society for Autism. Okay, the floor is yours. Please start to share your presentation, and after that we will open a discussion. In some previous webinars which we organized with Autism Research Institute, we have Macedonian translation, but now we will have you in live. Okay, please start.
So is it showing? Is my presentation?
Yes, everything is nice, and please start.
Okay, thank you. I just have to get over to PowerPoint so I can see what I'm talking about. There we go. Well, thank you everyone for joining me. I know it's late in the day for you and it's morning for me. And I appreciate the long time that we've known each other, Vladimir. And today I'm going to talk about the work we're doing on the gestational period and the influences that changes in the maternal immune system may have on the development of autism. As everyone is likely aware, there are many potential etiologies that have been proposed for autism. We have the skewed inflammatory sort of picture of cytokines and chemokines, and that is one of the things I will talk about today. Environmental insults, and of course, we always think about genetic predisposition that may be underlying things. People have discussed the microbiome, so the gut bacterial composition, infection, which also plays into the skewed cytokine profile potentially, and then lastly, the anti-brain antibodies or the autoantibodies that recognize proteins in the developing brain, which is my largest body of work. So today I will talk about the gestational immune dysregulation and the maternal autoantibodies. So there's been a lot of epidemiological evidence that implicates maternal infection as a risk factor for autism and schizophrenia. And I think the earliest literature actually was in schizophrenia, where they started to look at the season of birth. That was a time of flu and infections, and that this potentially could contribute to changes in neurodevelopment that led to schizophrenia. And now we've also adopted that potential model for autism. And as we know, when women are pregnant, our immune system changes greatly because now we have a foreign body in us and we have to protect that foreign body from rejection by dampening down the part of the immune system that would be a cellular immune response that might fight infection. However, sometimes we don't see that regulation occurring, and that is what has given us the maternal immune activation model. So as an infectious agent enters the body, it would stimulate a response, and that response is very common. But unfortunately, if that response continues, we increase our risk for altered neurodevelopment. So the idea is in the fever response, which is perfectly normal. However, in some women, this can be a stronger response or can go longer. So impaired immune regulation would be the reduced ability to control that inflammation, so that that inflammation does not damp down as it should, and you may see a change in the profile. We also can see increased inflammatory cytokine production. And I think this is the hallmark of maternal immune activation and that model. And so this is what we see in many women who have risk of a child for autism or a child at risk for schizophrenia. So underlying this, and a really important thing to remember, many of us get sick during pregnancy. We don't all have a child with autism or schizophrenia, is that underlying genetic susceptibility, or the genes that may control how you respond to an infectious agent. Also very important, and maybe also important in distinguishing schizophrenia from autism in this model, is gestational timing. So when during pregnancy does this infection or perturbation of the immune system occur, and the intensity of that immune response. As I said, if that immune response is long lasting, and if it goes higher than would be considered normal, that may be setting up the risk for altered neurodevelopment. And then there's always the possibility of other postnatal events that may contribute to this. So we have done some work on looking at the autism spectrum disorder and developmental delay without autism risk factors in a clinical population. This is what we call the early markers for autism study. This is work I've done with Lisa Krohn at Kaiser Research. And these samples were taken from a Southern California collection of samples that were banked between 15 and 19 weeks. And this was for the extended alpha fetoprotein screening program, which is a program looking for potential risk of altered development. And we took advantage of having those samples collected to create a screening assay. So these were collected between 2000 and 2003. So this is quite a while ago. And you can imagine the autism risk has gone from, I think it was 1 in 500 at that time, to 1 in, I don't know, it depends on which state you're in, but 51. And so I think we're at a point where we've seen such an increase in trajectory during that time period. So this is the window that these samples were taken. So this is early in the second trimester. And this is important because we think that its second trimester is a particularly vulnerable period for the immune dysregulation effects. So this study compared mothers of children with autism with no intellectual disability, mothers of children with autism with intellectual disability, developmental delay without autism, or intellectual disability without autism, and then a general population control, meaning they were never in the system for having a child with autism, but we can't rule that out, that they did have a child with autism. So what we really saw, and this is I know it's very data heavy, these are just the p-values of the changes. So just comparing this whole group, we have the growth factors such as GM-CSF, the innate inflammatory cytokines, meaning those come from the innate immune cells like dendritic cells, macrophages, monocytes. Th1 cytokines, these are the ones that would fight a viral infection. Th2 cytokines, these are more regulatory, they downregulate the Th1, but they also could promote allergy in IL-4. Th17 is also an inflammatory cytokine produced by a specific set of T cells. The receptor antagonist IL-1 receptor antagonist is actually a downregulatory, and you can see that is the only one that is down in this population over here. And then the innate inflammatory cytokines, meaning those, again, chemokines. And chemokines are the factors that tell cells where to go. So these are the ones that would call in cells during an infection. And as you can see, if we just look at ASD plus DD with ID versus GP, we see some elevated cytokines. If we have ASD with no ID versus GP, we don't quite reach statistical significance, but with interferon gamma, we see DD versus general, so developmental delay versus the general population, we see more decreases in the innate chemokines. And this is important because those same chemokines are what are used to direct the cells that are in the developing brain. But I'm going to jump over to the last column, and the biggest difference we saw, and I think one of the most interesting aspects of this, was that the mothers of children with autism and intellectual disability had elevated inflammatory cytokines and chemokines above all of the other populations. So, but also from ASD with no ID. So this profile separated them from all other groups, but also subphenotyping, or separated them from those mothers who had children with autism and no ID. So that intellectual disability is potentially associated with autism is potentially a result of this inflammatory profile, but it is not the same in developmental delay or intellectual disability without autism. And I think that is also a very important point. But it also suggests a lack of selective immune regulation. So that downregulation of the immune system, especially during the second trimester, is not occurring in this population. The other thing that we've done is looked at the child's immune system at birth. So we collect newborn blood spots, which you can see through a heel stick in the upper right corner. And this is a bank of these newborn blood spots for the newborn screening program that is kept by the state of California, and several states in the US do this program. And we use them to measure immune markers at birth. And the one thing that we need to remember is the child's immune system at birth is very, very immature, so that until you are born and start having exposures, your immune system, that is when it starts to develop its specific responses to things. So what we usually see at birth is a very dampened Th1 or inflammatory T cell profile, and an upregulation in the Th2 or sort of the more regulatory profile. And this is where chemokines are very important. And what we noticed in this study is that newborns that were later diagnosed with ASD and delayed development or have lower levels of cytokines and chemokines at birth compared to those with typical development. So remember when I said that these cytokines and chemokines are very important for getting cells where they're supposed to be, and these chemokines, especially in this early period in early brain development, are critical. And there's just a couple listed here that we saw, the CTAC and MPIF1, which are the strongest predictors of ASD compared to typically developing in DD, respectively, those CTAC for TD and the MPIF1 for DD. And that higher neonatal levels of the chemokine CTAC were associated with a 60% decrease in the odds of ASD relative to TD. So higher levels are protective. And I think that is a really important point. We often look for what is higher as a negative, as we were looking at in the mothers. But in the case of the newborns, those higher levels of chemokines that get the cells where they're supposed to go are actually important. And the higher levels of MPIF1 were associated with a 138%, which is high, increase in the odds of ASD relative to TD. So this chemokine differentiates ASD from DD, meaning that this is a chemokine that is even that is implicated in developmental delay without autism. And I think another area of interest is certainly we have just the higher number of boys to girls. That ratio is 4 to 1, and it's probably changing a little bit. But one of the things that we've looked at is how the child's sex affects their immune system profile at birth. And I think this is something that has not been looked at a lot. We tend to lump boys and girls together in many of the studies that we do and in the animal models that we do. But we noticed that the neonatal immune signature differed by sex, regardless of what that neurodevelopmental outcome was. The exception to this, so where girls had a little more active, actually, immune system than boys. And so the pink is the girls. We went very traditional here. Pink is the girls, the blue is the boys. And you can see this effect size, meaning how strong the effect was for a given cytokine or chemokine. And the farther it goes, the more it was. The stars indicate that these are significantly different, where MCP is lower in the boys significantly. And this is not anything to do with the outcome. This is really just what the immune system at birth looks like between males and females. But what we did notice, if we looked at a difference between ASD, developmental delay, and the general population controls, control males had higher macrophage inhibitory factor, or MIF, than the other two, than both females and than the other population. So meaning there's a deficit in females with ASD and DD and the males with ASD and DD. So that this appears to be an important factor in these two, in developmental delay and in autism. And again, that is a chemokine. So as I said, the chemokines are really important for healthy brain development. We can't infer any more from this study because these are newborn blood spots. We're not looking at brain, but what we can say is this difference could turn out to be important later on. So I'm going to switch gears a little bit and talk about another type of immune dysregulation during pregnancy, and this is maternal autoantibodies. So autoantibodies are antibodies to self-proteins. This is normally regulated by your immune system to not produce these. I mean, I'm sure everyone is familiar with autoimmune disorders such as systemic lupus, rheumatoid arthritis, myasthenia gravis, MS, multiple sclerosis. And I think, so knowing that we have autoantibodies that target proteins in the developing fetal brain, these are not fetal proteins, but they're very highly present in the developing brain. So we call this maternal autoantibody related autism, or MAR-ASD, which is a subtype of autism, in our opinion. That this is something that we discovered in our lab. And I think just to walk you through what the sort of how this might work, maternal IgG crosses the placenta during development, and that is really important to protect the fetus, right? That passive transfer of everything the mother's been exposed to and made a response to gets transferred to her fetus. In cows, or in horses, they do it through colostrum at the first feed, but in humans, we transfer, and rodents, they get transferred across the placenta. These antibodies can also access the developing brain because the blood-brain barrier is more permissive during this time period. So we've shown that they do get into the brain. Unfortunately, some of these antibodies, if they are responsive to self-proteins, can bind to their targets. So some antibodies can bind to these neural progenitor cells that are the early neurons in the brain and affect their migration, their maturation, what they look like, their anatomy, and their function. So this could lead to abnormal brain development, including brain enlargement, and the behaviors that we see with autism. So we did identify the target proteins for this. It was a very crude test when we first, but what we noticed was anytime we saw, if we have a brain, and this is a Western blot, if the brain is what we ran in the gel, fetal brain, and we probed it with antibodies from women with children with autism, typically developing or developmental delay, we really only saw this type of pattern in the autism samples. And this is just an example of one pattern. We went on to identify what those proteins are, and each of them, I'm not going to get into them specifically, except to say that they each have a role at during some point in neurodevelopment, and that includes neuritis, dendritic brain.
Џуди, your microphone, please. Your microphone, it's turned off. Please turn on. Sorry. We still don't.
Okay, is that better? Sorry.
Yeah, now it's okay.
I don't know how
Yeah, now it's okay. Your microphone was turned off for 20-30 seconds. Please go on.
That's very strange. It just turned itself off. I don't know why. So just to give you the end here is that each of these is important in some aspect of neuron development, and that the final sort of maturation point are CRIMP1 and CRIMP2 are responsible for. And if you remember these come up later, and their importance. And the other point is that this effect is additive, meaning reactivity to just one of these is, and patterns are two or more, and there's certain patterns that are very, very highly specific for autism. So what we did is we created an ELISA assay, enzyme-linked immunosorbent assay, and this is an assay that allowed us to use those proteins we identified to test for maternal autoantibody reactivity against each of these proteins. And it's a very simple assay to do, and we had 450 mothers of children with ASD and 342 mothers of children who were typically developing. We had a very large data collection, and it took a little bit of machine learning to determine which reactivity patterns were specific for autism. So we have a team of 4 statisticians on this paper that was published in 2021 and in molecular psychiatry. And it took a large team to make sure that we had very accurately assessed which of these patterns were specific. This is a very complicated table, but just to use it to show that the patterns the 3 top patterns that were most specific were CRIMP1 and GDA, CRIMP1 and CRIMP2, and NSE and STIP1, with some of the less frequent but still significant patterns down below. And this is mostly just to tell you that if you look at how many typically developing we saw, you can see the zeros in this column where all but these bottom 2 patterns were very specific, and we did not see them in the typically developing samples. Over here, these are the odds ratios, meaning what is the odds that you will have a child with autism if you have these present. We would have a higher odds ratio if we had even more samples, but this is still a very high. So 31 times more likely to have a child with autism if you have CRIMP1 GDA, and so on down the patterns here. So even if they're not specific, they still give you an increased odds of having a child with autism. So these are the top specific patterns, as I said, and this is what we are using to actually try to create a test for people. And then the other important thing that came out of this paper was if you had an antibody to CRIMP1, your increase in the ADOS severity score would go up by 2.3 points. So if you had an ADOS severity score of 6, it would be elevated to 8 if this pattern was present. The other thing we noticed is that there are some subphenotypes that I will talk about in a second that are associated even within this subphenotype of autism. So the presence of CRIMP1 increases the odds of higher ADOS severity scores twofold. So there is something very specific about CRIMP1 and CRIMP2 to some extent. So that was in samples that were taken at the time the child was diagnosed from the mothers, and that was what we call an associative study. But what we are really interested in is if they are present during pregnancy, would we be able to predict outcome? So we did, again, with Lisa Krohn at Kaiser, the same EMA study that you saw with the molecular, cytokine and chemokine that were specific for autism with ID. This is a mid-gestational maternal plasma, again, enrolled in EMA. We had general population controls, 420, developmental delay without autism, 184, and mothers of children with autism, 540 in this sample set. So we use the same ELISA, and then we use the same patterns that we had identified in the first study to see if they held up in this study and what the specificity of that would be. So in this study, we had fewer. This is an older sample set, which I think is an interesting part of this study. These, as remember, I said these were taken 2000 to 2003. The study in which we did the discovery work were samples that were up into like 2012, 2013. So already we had seen an increase. And in that population, it was about 18% were positive. In this population, it's about 12%. So what we did see, though, is the same patterns were showing up as highly significant, but a little bit different distribution. If you went from most frequent to least frequent, we dropped the pattern of STIP1Y box after this study as a pattern because it had little specificity. So just wanted to, so as we go through this, we're refining it even more. And then we saw that CRIMP1 and CRIMP2 was actually the major pattern and gave you an odds ratio of 15 for severity. So this was highly associated with ASD outcome and not developmental delay. So we did notice that that was certainly an outcome of this. And just to remember that the general population controls were not validated as typically developing. So it could be that there were some cases of autism that were missed, that because they never sought services in the state of California for autism. And the other thing I think that is important about this study is that these antibody patterns, so the certain patterns could predict whether the child was autism with intellectual disability versus child with no intellectual disability, or autism with no ID versus GP. So as I said, we have this subphenotype, even within this phenotype, of the pattern can be predictive of the autism severity and potentially what type of autism the child would develop. And I think this is an exciting part of this study for us, and we are continuing to look at this in additional studies, because clinically this might be quite useful. If you have a pattern, if the child has a pattern, or is associated with a certain pattern, what type of intervention and therapy might be beneficial for them. So for the clinical studies, just to conclude, we found that reactivity to any of the MAR-ASD patterns was highly accurate in their ability to predict autism in risk in both of the studies that I presented. Reactivity to CRIMP1, CRIMP2 was associated with more severe ASD with an ID co-occurrence. If you had antibodies to CRIMP1, you also saw more severe autism. And some patterns trended to be associated with an ASD with intellectual disability or autism with no intellectual disability. So hopefully, as I said, these will be useful to facilitate the appropriate early intervention services based on that child's specific needs. And we also saw that the patterns showed a different distribution depending on geographical differences between these two study populations. And in additional studies that we've done, we also see that there are different patterns and a different percentage of the population that is positive, a little bit depending on what geographic location they are from. And so I think we will end. This is the human clinical, and now I'm going to go to what we call our preclinical models. And an important question we always want to address is, are these antibodies pathogenic, meaning do they actually cause disease, or are they merely biomarkers that something has occurred and that increases risk, that event increases risk of having a child with autism? So one of the ways we do this, we can passively transfer human antibodies that are positive into animals. We've done that in non-human primates in 2 studies, and we've done it in mice. But more important, I think, for us was to actually create a mimic of what we saw in the human clinical condition and use an what we call active immunization to break tolerance, meaning you should be tolerant, your immune system should not recognize these proteins, and we want to push the immune system past that so that they now make these antibodies. And then we do this in the female animal. We've done it in mice and in rats prior to breeding. And so this is our current working model, and this is just a picture of how we do this. So we take the peptides that mimic exactly what the clinical samples react to, and we create the same antibody profile in the animal. And then once we've confirmed the titer, then we go ahead and we breed the animals, and then we test the offspring at birth for using many different metrics. Especially important is behavior, but we've also done some MRI imaging, MRS imaging, immunohistochemistry, and currently ongoing are the cell culture studies. But these antibodies are transferred to the offspring, and we can show that. And in both our mouse and our rat model, we saw altered social and self-grooming behaviors, and which is really good. We did the same model in both animals. Rats are a little more, they have a little more complex social behaviors. They've got some prefrontal cortex that a mouse doesn't have. So they're a little more a higher functioning animal model for us to use. But we did see a replication of the reduced behaviors and increased stereotypic behavior in these animals. So especially significant at this juvenile time period where the animals are incredibly interactive and do a lot of play, if you look at total play as the metric compared to adults. And I can play, so this is just a video of, you can see the arrows highlighting the control animal, meaning they just got saline plus the adjuvant. And there's a stimulus mouse that gets put in, and the stimulus mouse is there for this animal to interact with, to see how much they interact with each other. And the control animal is very social. Actually, it's a little bit more social than our stimulus animal in this case, but they do a lot of it's very basic, sort of the metrics are on the top there that we measure. And then you will see in a moment what the MAR-ASD animal looks like. And it is very easy to tell this animal apart from the control. And really what it's doing there is doing a self-grooming stereotypic behavior and actively avoids interaction by crossing to the other side of the cage to continue to do its self-grooming behavior. As you can, it looks like it's grooming its whiskers. And the poor little stimulus mouse goes over and tries to re-engage the MAR-ASD mouse, and is again sort of avoided and goes back across the cage so that it doesn't have to interact. And we've done the same behaviors in our rat. The rats are a little different, and they're very hard to score because they're very busy. But what you do see in these animals where the control animal, there's a stimulus animal and the saline control animal playing there, and you can see there's a lot of mounting behavior, but a lot of play in these animals, and they don't even leave each other. Now you can see, and you should be able to guess which is the MAR-ASD animal here, but it's the one with the dark mark on it, and it is not interested at all, does not engage, is completely paying no attention whatsoever to the other animal. And and it just stops and starts licking the cage. Different from the mice, the stimulus animal kind of gives up and doesn't try anymore to engage. So rats are a little more, and we saw the same thing in the non-human primates. So This we also saw, so we saw decreased communication in these animals, what we call ultrasonic vocalizations. We saw decreased social behavior. You saw a brief snapshot of that. And something that rats generally don't do, which is repetitive grooming behavior. And we saw an increase in that. And when we looked at the brains of these animals, so we did scans at postnatal day 30 and postnatal day 70, we saw that, interestingly, that the males and females, first of all, are different in these animals. The control is in the blue and the MAR is in the red. And you can see in the males, we see lower total brain volume. And the females, we see an increase in brain volume. And for some, there are relative volumetric differences in different regions of the brain, where we saw changes in sensory cortex and cerebellum in the males. In the female only, we saw changes in the midbrain, and then combined, we saw midbrain and cerebellum changes. So we do see differences in the way the brain is developing. We don't see differences in behavior between the males and females. So that's a really important point. We don't see a sex effect of these antibodies on the behavior, but we do see it on the way the brain develops. And so it's interesting that you can get these differences that don't translate to behavioral outcome. We also looked at the metabolite levels in the brain, and we saw, again, differences where we saw reduced choline compared to the controls, reduced glutathione compared to the controls, and increased taurine compared to the controls. When we do our maternal immune activation rat, we see very different profiles. So this is different than the maternal immune activation model, this autoantibody model. And it's possible, because pyruvate is part of that taurine pathway, that these antibodies to LDH may impact the lactate dehydrogenase enzyme, which converts pyruvate to lactate, may be impacting what we're seeing in the brain, because these animals did have these anti-LDH antibodies during gestation. So just to conclude, it was a lot of data, but the gestational immune dysregulation may contribute to the risk of altered neurodevelopment. And I think we're very careful not to say autism or schizophrenia, because what we're actually seeing is a change in the way the brain is developing with these inflammatory molecules in our animal models. This may be underlying mechanism behind the increased risk due to infections during pregnancy. The specificity, as I said, for autism may not be clear at this moment, since schizophrenia may have a similar risk factor. And it could be due to timing and genetics. You know, the genetic background could be the difference between those two. So in vitro studies demonstrate the effects of inflammatory cytokines on neuron health and development. And then we also, I talked about the autoantibodies, and these antibodies localize to the brain of the developing animal. We've shown that they actually go into the fetal brain. And we see enlarged brains in humans, in our human, clinically associated with these. In humans, in mice, in monkeys, and in rats. So I didn't talk much about the non-human primate today, but we do see with that passive transfer similar changes in the brain volume. And I think our active immunization model, mouse and rat models of this type of autism capture all three domains of autism, including the social aspect, which was very visible, communication, and then the repetitive behavior deficits. So I think, you know, we've come to the conclusion that MAR autoantibodies alter neurodevelopment, but it also, including total cerebral volume in the humans, monkeys, and the rodents, and demonstrates structural effects of these autoantibodies. So I think this is what we're working on now, is trying to understand those structural differences and how those directly impact development. So I have a great team of people and a great group of collaborators. I've recently graduated at 3 of these students. I have a new student who hasn't worked, is just getting started, but Matt did the rat model along with Jana, who's continuing that on. Ale is the one who did all the human clinical studies. Danny did the newborn blood spot studies. And Chelsea is working on creating this test in newborn blood spots where we can test for these autoantibodies. and is also working on our maternal immune activation models in monkeys and rodents. And then Melissa Bauman with the behavior. Paul Ashwood is an immunologist who trained under me as a postdoc and is now a faculty at Davis. And then Lisa Crowan, who Kaiser, when I talk about the EMA study, the Kaiser research. So thank you, and I'm happy to take any questions. I'm going to stop presenting so that we can communicate here.
Yes, thank you very much, Professor van der Water. It was really incredible presentation. I really like, and also I think that the participants who were involved in this webinar today, it was our 23rd webinar in last 2 years, 2.5 years, which we organized, and it was incredibly very, very good. So also I'm enjoying reading your papers of you and Dr. Paul Ashwood, Dr. Lisa Crowan, etc. So I hope that our collaboration will continue in the future. We are waiting for the questions in Q&A session. So please be free to put your questions in the box. Also, I would like to say that you are invited to write some paper on this if you have some unpublished data. In my new journal, I wrote something about that by email 2 weeks ago. Mm-hmm. So if you have ambition and if you have time to write something, some review article, because we need professors like you to publish at least one article for us to have a really good international journal, because we started last year with colleagues from Slovenia. Macedonian Scientific Society for Autism is also a co-publisher. The main publisher is from Alma Mater Europeia University in Maribor, Slovenia. Okay, then I will go to the first, if you don't mind, I will go to the first question from Irena Stojanovska. She was my master student, and she got a master degree last year on special education and rehabilitation on autism and intellectual disability. She is asking you, what causes other than infection during the gestational period can initiate the inflammatory maternal reaction with perturbed antibodies cytokine chemokine profile that might affect fetal neurodevelopment? Maybe maternal autoimmunity, environmental triggers. Also, in my handbooks, which I wrote on Macedonian, I cited most times your articles from early 2000s, and a lot of my students are familiar with this, but the medical doctors are not so very familiar in Macedonia, because also it is I'm very sad, because today we have only 37 participants and 102 people were registered on Google Drive form, but it's probably it's evening and they're very busy or very tired from the
stuff happens. Yep.
Yeah. And Macedonia is a small country, probably you know, we have less than 2 million inhabitants.
But
okay, you can also read the questions if you don't understand me well. You can read in the Q&A session.
So where? Oh, there it is, right there. Okay, Q&A. Let me get
Irena Stojanovska. I read it for you, but if you don't understand me well.
Okay. Yes, it's
what
easier. What
causes other than infection?
Yeah. What causes other than infection during gestational period can initiate the inflammatory maternal reaction? I think that's a good question, and I think anything in which the mother does not downregulate effectively their cytokine chemokine profile, right? So, as I said, during pregnancy, the normal sort of physiology is to have a more regulatory profile because you have you don't want to reject the fetus, and sometimes you will have an elevated cytokine profile, meaning you're not downregulating effectively. So it's more of a regulation issue than a response to infection issue that can then change the cytokine profile during development. And then I think it is a little bit, and one of my students, Jana, the redhead in the picture, is actually looking in EMA, since we have cytokine and we have the autoantibodies. She's just finishing up a paper on that and comparing those two to see if there is a cytokine profile that is related to the production of these autoantibodies or just any of the 8 autoantibodies without a MAR-specific target. And it is looking like that the MAR autoantibodies have a very different maternal immune profile than just production of any autoantibodies. So stay tuned for that one. But maybe maternal autoimmunity, environmental triggers. Environmental triggers are important. I think it's another area of interest for us. We have a big environmental health center at Davis, and the question is, yes, some environmental exposures can cause changes in immune function.
Yes, of course, especially polybrominated polyurethanes. Yes, you mean about that, and chlorinated chemicals. Yes, I read a lot about that, and I appreciate very much your team, and we saw also young colleagues. And I hope that it will be a strong impulse to my younger colleagues who were present on this presentation. And I strongly believe in your theory and about maternal immunity, autoimmunity hypothesis of Dr. Singh Viendra and all of your researchers in United States, and also in my PhD, which I worked, as I said in the beginning, from 1999 until 2002. We also in Macedonian population, we prove the immunological theory and autoimmune hypothesis in our sample. We found IgG4 increased in children with autism. We found specific allergic antibodies. Unfortunately, we did not maternal autoantibodies. In that time, we don't have so much equipment to do that, but a lot of autoimmune theory and immunological theory we proved in Macedonia, and also our team published a lot of articles on English, and you can find them. Okay, we have another one question from Irena. She's very good young researcher, and she's asking that your research focuses on the effects of maternal autoantibodies on altering fetal neurodevelopment. Do you have any insight whether the child's own autoantibodies in children with autoimmune predisposition can also adversely affect postnatal brain development, possibly in the form of autoimmune-driven encephalopathies?
That is a great question. We did a long time. So I can tell you that the children do not have the same autoantibodies as the moms, so that we have looked at. They have their own potentially set, and that's something that we're actively looking at right now. The maternal autoantibody story is a little more mature because there was greater specificity that we saw in those, but I think there's always the big question in any autoantibody-driven pathology that is associated with the brain is that ability of those antibodies to get to their targets in a more mature brain, right? And during that early period of gestation, that the antibodies the blood-brain barrier is more permissive to antibodies getting across. The other thing we're looking at, not just brain, is antibodies to the GI tract as a potential immunopathology. So I think stay tuned. That is something that we are actually doing in the lab right now. I think people are a little less inclined to think that the antibodies can be pathogenic, and probably more inclined to think they might be biomarkers, that something has happened. But we will see, because that is something then we can create, just like we did in the maternal story. We can create the animal model by creating those autoantibodies and seeing what happens if the child, let's say a child regresses. Or the offspring, sorry, the offspring.
I have two questions also for you. Do you believe in the regressive hypothesis of autism? And what is your opinion about connection in gut-brain hypothesis and hypothesis of Dr. Andrew Wakefield about immunopathology of lymphoid cells in the gut, and also something about that. I strongly believe that there is connection between gastrointestinal pathology such as obstipation, diarrhea, gastroesophageal reflux, etc., and behavior of children with autism spectrum disorder.
That was a very big question.
Yeah, I know, but in the short, I know, but because the people are mostly special educators and psychologists here today. I see probably only one student on medical faculty, and I'm not sure do we have another, and some parents. So shortly, and then we will close the webinar. It's around 8 o'clock here and 11 there.
Yeah. So I think to answer your question, regressive, I do think there are kids who have, because we see so many forms of autism, absolutely there are kids who have met milestones and then lose those skills. What causes that regression, I think, is a huge question. Is it just, that they had sort of the basic, and that something then as the brain continues to develop, something is at play and the brain then begins to not develop normally? What causes that? I think that's a huge black box. I think the Andy Wakefield story is I think it wasn't a complete story, and I think that's a problem. That caused a lot of problems. And I think, as scientists, very data driven, and you need to prove something to me, over and over again. Okay, we can potentially accept this. I mean, the maternal autoantibody story was not accepted at all for a very long time. So I think that is something that we continue to look at. What events may have occurred, what exposure. It may not be vaccination at all. It could be some other exposure, during that time period. And then someone asked in the chat, can blood from newborns be used?
Please read it. I saw it now.
As early markers of ASD. And I think that is the hope. So I alluded to some work that we're doing with the newborn blood spots. And the other thing is we're developing the maternal autoantibody test in the newborn blood spots so that it could be like a screening. And we're hopeful that that will give us more information. So I think the problem with cytokines and chemokines as a marker is they are markers for lots of things. And so you're sort of limiting yourself it's not quite as specific enough, but if showing that you have a lower level of something that we think is very important for especially trafficking of cells, then we potentially could use that as a marker. So that, that is always the hope that we'll be able to do that.
Yeah. Okay. Thank you very much for your very good explanations of our questions and especially of your presentation. I'm very glad that you accept my my offer immediately when I sent to you the email. So we are open. Me and our association is open for future projects. If you need some control group from Macedonia, which we have some probably other immunogenetic structure rather than American population, and so if you need that also in publishing papers and some projects, we are open for collaboration. Thank you for being with us. I wish you to stay healthy and to stay enthusiastic with your research, because you research more than 25 years and probably more in this field, and we appreciate your articles, your projects, your experience. So thank you in front of Macedonian Scientific Society for Autism and our membership, and also parents of children with autism from Macedonia. Thank you very much, and hope to see you probably sometimes on some other webinar, or if you're willing, we can invite you next year probably with some other topic.
Absolutely. It's a pleasure, always a pleasure. And thank you so much for your support in the work that we're doing.
Yeah, thank you also for the participants who spent 1 hour with us in this evening hot hours here in Macedonia. It's still summer, and so I want to say to all of you to stay healthy. The COVID is all around us again, unfortunately, but we hope that we will survive this this again. Yeah. Because we have a better immunity in comparing in 2020. Okay.
That and a less destructive strain, I hope.
Yes, a less destructive strain of coronavirus. Yeah.
Virus. Thank you, everyone.
Okay, we will see with our colleagues probably in October or November with another professor or doctor or special educator. We'll see who will arrange the next one. Okay, bye and have a nice day in USA, and have a nice evening for Macedonian and Balkan participants. We have one participant from Serbia today. I see in the list of participants.
Wow.
Okay. Yeah.
Thank you, everybody.
Yeah.
Bye-bye.
Bye-bye.